Zinianz research video. Liver is an important organ actively involved in metabolic functions. It is a frequent target of number of toxicants. The purpose of this study is to investigate protective effect of resveratrol against carbon tetrachloride-induced oxidative stress in rat liver. Carbon tetrachloride is a hepatotoxin, causing liver necrosis, fibrosis and cirrhosis when administered sequentially. Resveratrol is a non-flavonoid polyphenolic compound that is found in a great variety of plant species, some of which are part of the human diet. These include peanuts, grapes and red wine. It has several pharmacological activities including anti-cancer, cardioprotective, neuroprotective, antioxidant and anti-inflammatory. Method: pathogen-free male Worcester rats initially weighing 100 to 150 grams were procured. After acclimatization, rats were randomly divided into 4 groups each consisting of 10 rats. Group A, normal control, animals received vehicle only, 0.1 milliliters of mineral oil, orally. Group B, carbon tetrachloride control, animal received carbon tetrachloride, 0.4 grams per kilogram dissolved in mineral oil, intraperitoneally. Group C, treatment group, animal received resveratrol at a concentration of 100 mg per kilogram, orally along with carbon tetrachloride, 0.4 grams per kilogram. Group D, treatment group, animal received resveratrol at a concentration of 200 mg per kilogram, orally along with carbon tetrachloride, 0.4 grams per kilogram. Experiment was conducted up to 8 weeks. At the end of this period animals were sacrificed. Blood and liver samples are collected for biochemical and histopathological examination. Results: histopathological examination. The histopathology of liver from Group A showed a normal arrangement of hepatocytes with clearly visible nuclei and central vein. In Group B, intraperitoneal injection of carbon tetrachloride showed extensive centrilobular necrosis around the central vein of liver and dilation of the central vein. In Group C, the magnitude of dilation of the central vein was prominent but very less in comparison with the carbon tetrachloride control. The liver sections from the Group D showed good protection of hepatocellular necrosis with a poorly dilated central vein and regular arrangement of hepatocytes around the central vein. Very few necrotic zones were prominent. Biochemical levels. Administration of carbon tetrachloride to rats caused significant increase in serum enzymes such as SGPT, SGOT, alkaline phosphatase and bilirubin, and decrease in serum total protein level and albumin level compared to normal control rats. When rats treated with resveratrol 100 mg per kilogram orally, exhibited a significant hepatoprotective effect by reducing the serum bilirubin level and enzyme levels, and increasing the protein levels compared to carbon tetrachloride-induced rats. Resveratrol 200 mg per kilogram treated rats showed more decrease the levels of serum enzymes, and increased the levels of total protein and albumin compared to resveratrol 100 mg per kilogram, and carbon tetrachloride control rats. Lipid peroxidation. The degree of lipid peroxidation was measured as one of its end products, malondialdehyde. Carbon tetrachloride intoxication significantly increased the liver content of malondialdehyde. Treatment with resveratrol 100 mg per kilogram and 200 mg per kilogram showed decreased levels of malondialdehyde. Total antioxidant. High levels of free radicals are generated when treated with carbon tetrachloride. For this reason very less amount of total antioxidant, sometimes complete absence, were seen in carbon tetrachloride control rats. Total antioxidant levels are very high in resveratrol 200 mg per kilogram because it acts as a potent antioxidant agent. Glutathione levels. Glutathione levels are very less in carbon tetrachloride control group. These levels are gradually increased, with resveratrol and resveratrol-treated groups. Conclusion. In present study the data first indicates that chronic carbon tetrachloride treatment provoked a clear toxicity in rats. With the analysis the authors conclude and confirm protective effect of resveratrol and its antioxidant activity in carbon tetrachloride-induced liver injury. Zinianz. Control. Cater.